Good afternoon, everyone. Thank you for being here on the afternoon of the last day to see me speak. I'm very excited to talk to you about one of my PhD projects on the effect of temperature on the development of hybrid and non-hybrid mound building wood ends. So as climate change worsens due to human impact, hybridization rates are expected to increase, which can be a good thing because of adaptive introgression, which is what happens when there's introgression of adaptive genetic material between hybridizing species or populations, such as has been very recently described in the Gulf killifish. So this fish exists in the Gulf of Mexico, which is heavily polluted due to industrial activity. And the way that populations of these species have found to adapt to the pollutants in their environment is by hybridizing with their sister species, the Atlantic killifish, which is adapted to the same pollutants. However, we know that there are also costs to hybridization, such as hybrid inviability, which is what happens when hybrid offspring fails to develop into adults, and that's been described, for example, in Drosophila, so hybridizing between melanogaster and simulange, where it's caused by a cell cycle regulation gene. And there is also hybrid breakdown, which is what happens when the fitness of a hybrid individual decreases past the F1, as has been seen in African cyclids, and it's caused by genomic incompatibilities in that example. So given this, it's very important to ask, under climate change, do the benefits of hybridization outweigh its costs? And a great system to study this are the hybrid mound building wood ends. So this hybrid system, these hybrids are formed by the two parental species, Formica polyctena and Formica aquilania, which are divergently adapted to temperature. So Formica polyctena has a more southern distribution that you can see on the distribution map in yellow, and Formica aquilania, and it's adapted to heat, and Formica aquilania has a more northern distribution, they're in green, and it's adapted to cold. These species diverged recently, around 500,000 years ago, with asymmetric gene flow from aquilania into polyctena. And hybrids have been described in southern Finland, that little circle there, even though they overlap for quite an extensive part of their range in Europe. And we know that hybridization between these two species is likely to be recent and younger than 120 years, and that the genetic contributions of each parent into the hybrid populations are pretty balanced at 59% from polyctena and 41% from aquilania. And if you're wondering if hybridization is beneficial or deleterious in these ends, then you're in luck, because that's the next slide. So is hybridization beneficial or deleterious? We'll start with thermal resistance. So here we have results of a heat knockdown experiment that was performed at 46 degrees Celsius. And on the bottom of the plot, we have time in minutes. Then we have the percentage of surviving individuals on the y-axis, so at each time point. And the green line is aquilania, called adapted parent. Yellow line is polyctena, warm adapted parent, and the orange are the hybrids. And what this shows is that there's a significant difference in the thermal resistance of the two parents, and there is no significant differences between hybrids and the heat-adapted parents. So the hybrids survive as long as their heat-adapted parents under high temperatures, and that's great. Then, moving on to the standing genetic variation, in these plots, we have changes in allele frequency in hybrid males, which are haploid and therefore more exposed to selection. In both of these plots, we have the temperature in April on the bottom and the change in allele frequency on the y-axis. And what we see here is that so for you, it's the right-hand panel, the frequency of cold-adapted alleles increases in colder years, so towards the left of the plot. And in the other panel, we see that the frequency of the heat-adapted alleles increases in warmer years towards the right of the plot. And so this means that hybrids maintain the standing genetic variation of both of their parentals, and that they are able to respond to very quick changes in environment. But we also have evidence of hybrid incompatibilities. So here we have a circus plot of the hybrid genome where the chromosomes are all aligned in a circle. The red lines are barriers to gene flow, and the black lines that sort of link different regions of the genome are candidate incompatible regions that are in ancestry disequilibrium. And we also know from data from so developing individuals and adults collected in the wild, these are hybrid males as well, that there are genotypes that fully disappear between developmental stages and adult individuals. So life stage is on the bottom, then allele frequency, and we see that all of these alleles here, and these are macrosatellite alleles, disappear completely in adult hybrid males. So there's selection against incompatible genotypes during development in the hybrid males. So we know what happens with adult individuals, but how do the costs and benefits of hybridization change under different environments during development? And for us to be able to study this, we need eggs and larvae. So we got queens, we had them lay eggs under different temperatures, and this addresses the question of how does temperature affect the reproductive output of hybrid and parental queens. And then we let those eggs develop into larvae as well under different temperatures, and that answers the question of how does temperature affect the development of their offspring from egg to larva. So, if you remember from the distribution map, I told you that the hybrids exist there, and we went to some hybrid and parental populations that we know, but only for the Ephiculani parental, so the cold adapted parental, because we can't find currently pure Polyctena populations in southern Finland. We sampled 3 hybrid populations, 3 Echilani populations, and this is the very southern tip of Finland, and it looks very small. So if we zoom in, we see all of the Echilani populations are the triangles, and then the little dots are all the hybrid populations, so they exist very close together. So we sampled in total 135 Echilani queens, which means that we reached our goal of 35 queens per population, so we're very happy about that. Sampling of hybrid queens was much more challenging. In total, we managed to get 88 queens, and very unevenly distributed from each population, 47 from one population, 31 from another, and 10 queens from the final population. In total, we had 223 queens in this experiment, which meant I had a lot of nest boxes to manage. Okay, so we know our samples, and the way that we treated them after we collected them was we subjected them to 3 different temperatures, so 15 Celsius, 22 Celsius, and 30 Celsius. And here I'm showing the way that we actually did the experiment for only one temperature, but it was the same across temperatures. So a queen could either start her egg-laying assay in a sequencing petri dish, that's marked with an S, or in a viability selection petri dish that's marked with a V. And we did 72 total hours of egg-laying, broken into 6 periods of 12 hours, and every 12 hours we would switch them to the opposite petri dish. Eggs that were laid in sequencing petri dishes were collected for sequencing purposes. This is why it's called a sequencing petri dish. And eggs that were laid in a viability selection petri dish were allowed to develop into larvae under the temperatures at which they were laid. So, with our egg-laying data, we saw that temperature has a positive effect on the reproductive output of parental queens. So here in this plot, we have temperature on the bottom, the total number of eggs laid after 72 hours on the y-axis, and species on top. So the parental species is on your left, and the hybrids are on your right. And what we can see here is that Ephiculani queens lay more eggs than hybrid queens. So average over temperature, they lay about 2.4 times more eggs. And if we look at specific temperatures, we see at 22 degrees that they lay, I can see this is very small, about 3.4 times more eggs than hybrid queens, and at 30 degrees they lay about 2.6 times more eggs than hybrid queens. And we also see that temperature has a strong positive effect on Echilani queens. So comparing the egg-laying rates between 15 and 22 degrees, at 22 degrees they lay about twice what they laid at 15 degrees. And this was very surprising for us because we expected the cold-adapted parental to be much worse at higher temperatures than hybrids. Then when we looked at the hatching rate data, we saw that temperature has a negative effect on offspring development. So here we have species on the bottom, temperatures on top, and the hatching rate on the y-axis. And a very glaring result here is that eggs do not develop at 15 degrees, not even Ephiculani eggs, which is the cold-adapted parental species. We didn't find any statistically significant differences between Epheculonia and the hybrid eggs, but average over species, we saw 69% reduction of the hatching rate at 30 degrees compared to 22 degrees. So altogether, what this tells us is that the benefit of hybridization is not constant throughout the life cycle of hybrids. So if you're an adult hybrid, then you benefit from hybridization because temperature doesn't affect your reproductive output, you have a higher heat resistance, and you maintain the genetic variation of both of your parentals. And that is useful to you under different environmental conditions. But if you're a developing hybrid, then you do not benefit from hybridization because your survival during the first stage of development is severely decreased under high temperatures, and if you carry incompatible genotype combinations, then you're selected against. So what's next? One question that arose from our hatching data is, does environmental change affect selection against hybrid incompatibilities? And to address this question, we're going to look at genomes of the offspring of hybrid queens only by doing pooled sequencing of eggs and larvae. So we have here our two environments on your left. So 22 Celsius and 30 Celsius. We have data from two of the hybrid populations, and we will sequence pools of eggs and larvae from the same mothers, basically. And we know where the candidate incompatible regions are located in the hybrid ants' genomes. So we can go to those locations and look at changes in allele frequencies between egg and larva pools, because we expect that frequencies will be pretty similar in egg pools, because they have not undergone selection yet. And then in larva pools, there will be a change. And we're doing this under the hypothesis that allele frequency changes between egg and larva pools at candidate incompatible regions are different under different environments, which would indicate a genotype by environment interaction and would mean that hybrid incompatibilities are environment dependent. So I would like to just finish by thanking my supervisors, Vitor Souza and Jono Kulmuni, and everyone that helped me carry out this experiment. So Patrick Heidbreder and Lucas Broghart, who did field work, Sara Yamza, who did field and experimental work, and Patrick Kraft and Anna Carellos that helped me in the lab. And thank you for your attention. If you have questions, I'm happy to take them.
My name is Tianzhu. I'm a postdoc currently working on chromosome biology at Cornell University. And this is a project I did during my PhD, working on hybrid incompatibilities in butterflies. And so I think one thing interesting about speciation is that it's a complicated process, but it has some simple rules. And if you find simple things in complicated process, then it's most interesting because it suggests something might be common. And one of the so-called speciation rule in literature is a so-called Holden's rule. And I guess most of you have heard of it, which means if you cross different species together, and in hybrids, it's typically the sex with two different sex chromosomes. So either the males with XY or female with ZW chromosomes tend to be unfit. So sometimes tiger mate with lions in the zoo, and in those cases, the F1 male is always sterile, and F1 female is usually fertile. And the second speciation rule is first discovered in Drosophila, and is called the large Z or the large X effect. And so in this figure, on the x-axis, you're looking at the fertility of backcross in hybrid males. And the y-axis, you're looking at where the introgression occurs. So the top column, you're seeing the introgression, no introgression on the X chromosome. And when you have introgression on the X chromosome, then it leads to way more hybrid sterilities in the males. So the pattern has been pretty consistent in many different taxa. And this phenomenon is being referred to as a large X or large Z effect. And the classical explanation for these two rules is so-called dominance theory. And the key assumption is that the X chromosome or the Z chromosome, it has recessive alleles that can be exposed in the hybrid background. And these recessive alleles will interact deleteriously with the autosomes in the hybrid F1. But in a homogametic sex, it has two X chromosomes. And so these recessive allele can be sheltered by the dominant allele from the other species background. So it gets sheltered, so there's no incompatibility. The nice thing about dominance theory is that it can be applicable to both XY and ZW taxa. So our motivation is that most of the empirical evidence supporting these theories are mostly relying on XY males, which always disrupt male-specific traits. But there's almost no study on ZW taxa, which disrupt female-specific traits. So we picked the butterfly system because it's really easy to pair them, even if they don't want to mate. You just grab a female, grab a male, and plug them together, and it mates. So we're focusing on a pair of sister species. One is distributed in the Northeast Asia, one is distributed in the Eastern Asia. And across the complex, you can see that if we cross different species, the hybrid female is always sterile and the hybrid male is always fertile. So they conform to the Haldane's rule. So we just want to describe a little bit in more details what defect we see in F1. So in addition to fertility issues, we discovered that there's also growth problems in the hybrids. So the females, depends on how you cross the two species, are either too large compared to the pure species or too small. So these are the extreme individuals that you see, that they're really different in size. And because the hybrids phenotype in the females are different depending on the cross directions, so there might be asymmetrically inherited genetic elements here. So we just approached the problem with a backcross design for QTL mapping. So we, again, because the F1 males are fertile, so we can cross the two species, generate F1, and cross the F1 to the parental species, and then sequence the whole pedigree, and then determine the phenotype, and then do a QTL mapping. So here's the results. Trust me that this display problems, lot scores here. So the x-axis, you're looking at 29 autosome and a Z chromosome. And the y-axis is showing the lot score for the weight in the backcross. And you have to believe me, there's no significant peaks on the autosome, and there's a huge peak on the Z chromosome, which explain over 50% of the phenotype variance from a single locus at the center. And for ovary dysgenesis, we discovered there's no major additive locus across the genome, but there's a pair of loci about a quarter from each end of the Z chromosome, which interact epistatically and explain over 50% of the phenotype variance. Okay, but I'm going to be a bit more contentious and say that this is actually the incorrect interpretation. What we think is real is that there are numerous additive factors scattered both on the Z chromosome and the autosomes, and each of them have very small phenotypic effects. And these factors are interacting between the Z chromosome and on the autosomes. And why we think it is real? Well, the first evidence is that you can actually predict the phenotype in a backcross using only the proportion of introgression on the Z chromosome. The emphasis is proportion here. So for instance, for body weight, you can see that the female pupil weight in the backcross is almost a monotonic function with how much introgression they have on the Z chromosome. And ovary dysgenesis, you can see that with intermediate levels of introgression on the Z chromosome, it leads you to the best phenotype. And if there's no introgression on the Z chromosome or complete introgression on the Z chromosome, it always leads to complete dysgenesis. So the key is that no matter where the introgression occurs on the Z chromosome, as long as there's a fixed amount of introgression, it gives you a fixed kind of phenotype. And again, because we have this significant lot peaks, so we find that if we apply this infinitesimal model, it actually recover the QTL peaks that you see in the backcross design. So here I'm doing a bit more quantitative analysis. On the x-axis, you're looking at the marker position on the Z chromosome. And the y-axis, you're looking at the predictive power of using one marker versus using the Z chromosome ancestry. And the spiky thing is the prediction under one QTL model. And the dashed curve is what you should expect to see under a polygenic model. And you can see that the observed curve is closely follow the polygenic predictions. And we can use the basic logic, same logic to recover the same epistatic loci on the Z chromosome for the other phenotype. And another evidence is that there's interaction with autosomes. So I'm just going to draw your attention to this bottom corner in the figure. So these Z chromosomes have no introgression, but because they're analyzed in a backcross, so the autosome have, on average, 25% of introgression. So these defects in ovaries are actually caused by 25% of introgression on the autosomes. And we reanalyzed the only other such study in butterflies in heliconias from the same lab, and we more or less discovered the same qualitative relationships in ovary dysgenesis. So again, going back to the infinitesimal architecture, we think there is a balancing interaction between how much introgression you have on the Z chromosome and how much introgression you have on the autosomes. It predicts Haldane's rule because in F1 homogametic sex, say if they have 2Z or 2X, then have 50% introgression on Z and 50% of introgression on autosome. So there's some likelihood of being balanced. And in the heterogametic sex, because the Z can either be 100% from one species, or 0% from that species. So it's more likely to be imbalanced, which generates Haldane's rule. And it predicts large Z effect in backcrosses because usually there are way more autosomes than the Z chromosome. So the proportion of introgression as shown in blue is on average about 25%. But on a single Z chromosome, it can be highly variable. So if you're only looking at the backcross, then the variation of the imbalance is mainly attributed to the variation of ancestry on the Z chromosome, which leads to a lot score peak on the Z chromosome instead of the autosomes. And also predicts a large Z effect in introgression lines because if you introgress a fixed physical length of DNA, it will perturb the ancestry on the Z chromosome by a much larger amount than the same perturbation on the autosomes. So I just will conclude my talk with some of the remarks. So the model we propose is completely different from the dominance theory. As you can see, there's no mention of dominance. But it's equally applicable to the XY or ZW taxa. And if you trace back to the theory of Haldane's rule and related experimental evidence, Hermann Muller in 1942 proposed so-called imbalanced gene expression between sex and autosomes. And we think that our framework is actually very similar to his verbal arguments. And even though Muller is usually viewed as a predecessor of the dominance theory. And we also discovered recently there is a theoretical work by Thomas Lenormand and Denise Rose on bioarchive. So if you want to check that out, I would recommend you to do. And some take-home message. So first, which is also a surprising discovery myself, is that it's really non-trivial to interpret QTL results if you're crossing the pretty divergent species. And when you see a single peak, it could be many different things, depending on the genetic architectures. In our case, it's completely polygenic on the sex chromosome. So when you detect a major effect QTL, sometimes explaining over 50% of variation, in our case, it's actually more congruent with a smear of polygenes across the chromosome. And we have developed a theory to understand more in detail of why we get these artifactual QTLs. So if you're interested, please check out the supplementary information of our paper here. And with that, I would like to thank many contributors to the work. Sri Harsha, who took most of the phenotypes of the ovary, Neil, my advisor Jim, and the two collaborators, Xueyuan and Masaya from Japan and China. So we will have plenty of time for questions. Yes.
Thank you very much. Great presentation. I was just wondering if the phenotypic effects that you see in the hybrids, if you know if they present them if you cross individuals from sympatry or from allopatry or for both of the distributions of the species.
So we cross the individual actually from pretty geographically isolated, far apart regions.
So only for the allopatry?
Yeah, only from distant end of the distribution.
Okay, and do you know if when you cross from sympatry, these consequences are also seen?
So we haven't done the cross with individuals in symmetry. They're actually a bit parapatrically distributed. So we don't usually capture the same, both species in the same locality, but they are pretty much, I would say, like adjacent to each other, and they're separated by a really developed region which you don't see those butterflies at all. Yeah, but maybe there will be some differences, but they're pretty divergent from each other. So the DXY is about 6% on their autosomes. Yes. Yes.
Just fantastic. I'm very interested. I'm not a modeler, but I'm very interested about what's your opinion about. So now there's a lot of simulations demonstrate that a simple like allele mechanism of genetic incompatibility cannot maintain species boundaries
Mm-hmm.
because selection can just purge them out of the population easily. But for your finding, which is proposing the infinite, like polygenic basis of genetic incompatibility, how would that affect,
Introgression.
yeah, introgression or maintain of species boundaries? Like, because my thinking is like selection, it might be harder for selection to completely purge them out completely, those alleles. I don't know.
Yeah, I think if we trust the the ancestry balancing mechanism, even into the advanced generation of back crosses, we don't have any evidence for these because we only but I think it will lead to different dynamics. Thank you.
Hello, everyone. My name is Dmitri Dedukh, and I would like to tell you about cytogenetic mechanisms of asexual reproduction in hybrid loaches. As you know, interspecific hybridization have plenty of outcomes in hybrids. On your right, you can see that the most widespread outcome that usually hybrids have low viability or sterility. And sterility usually is caused by problem of different chromosomes coming from parental species to pair. On other extreme, on the left, you can see another situation. When pairing of orthologous chromosomes from parental species is possible, it can lead to genomic introgressions and also intermixture of both parental genomes, leading to something new, genetic combination. However, there is like another outcomes between crosses of two different species, which are much less studied, but still they are existing in nature. And this is emergence of so-called clonal or asexual hybrids. Those hybrids have maintained heterozygosity throughout many generations and exploit different than sexual reproductive modes, sometimes using other sexual species for their reproduction and sometimes not. There are a variety of different asexual or clonal, I basically equal these two terms, in vertebrates, in hybrid vertebrates, and some of them you know very well, such as parthenogenesis, but there are also gynogenesis, hybridogenesis, and much more less common. And today I will focus about gynogenetic reproduction. So basically, during gynogenetic reproduction, diploid hybrid female produce egg, which have the same genetic composition of the somatic cells of the mother. And these cells use sperm of any other sexual species just to activate this egg, but do not incorporate genetic material of this sperm. And these activated eggs results in an offspring, which basically are the clone of the mother. And I would like to, I'm interested in the mechanism of how those eggs are formed. And for this, I focused on two hybrid species complex of loaches, one in Europe and another one is in South Korea. And I will start from Europe. So basically, we have two sexual species, like orange and blue, which cross together and form a hybrids, diploid ones, where males are always sterile and females are able to reproduce clonally in nature population. However, occasionally, like even in diploid eggs can be fertilized by sperm of the sexual species and triploid progeny results. And in triploid, there is a similar situation. Triploid males are sterile, and triploid females are able to reproduce clonally. To study their gametogenesis, mostly I focused on two meiotic stages of pachytene and diplotin. And when I investigated the diplotin chromosomes of triploid hybrid individual, I found that there are even more chromosomes than expected, 2 times more chromosomes than should be in somatic cells. So basically it means that to form this egg, it was an event which called premeiotic genome duplication, which occurred somewhere in somatic cell. In germ cells, but before meiosis. But when I looked on pachytene stage, the little bit earlier stage, I found 2 different types of cells. First cells on the left, they basically similar to those I found in diplotin with full pairing and everything is fine and they have duplicated genome. However, another cell population, which is on the right, exists as an initial ploidy level and those cells have problem with pairing, so they have like some chromosomes are paired and some chromosomes remains as unpaired, like green you can see here. And interestingly, that these cells with initial ploidy genomes, which have a lot of problematic pairing, it's the vast majority of those, like of all pachytene cells. Further, we decided to investigate what's going on even earlier in germ cells. And we used cytogenetic markers which allow to identify individual chromosomes. So basically, by analysis of germ cells, like by counting dots, we can identify the ploidy of the cells. So in sexual species, we see two dots, so basically it means that they're diploid. However, in triploid hybrids, the majority of cells was triploid, so we observed 3 dots, and individually we observed cells with 6 dots, which basically are hexaploid, so it's confirmed our meiotic analysis. In diploid hybrids, we see the absolutely similar situation. So here, there is a summary of the reproduction in those diploid and triploid hybrids. So the vast majority of cells, like I mentioned here, the pink one, they exist with initial ploidy level. They're dividing, and they're the most numerous. They're going to meiosis, to pachytene stage, but because of problematic pairing, they cannot overcome the pachytene checkpoint. While the other type of cells, which marked as a green one, they're able to duplicate their genome, go to pachytene, and because of this premeiotic duplication, each chromosome can able to find a copy to pair. So that's why they don't have pairing problems, and they go through meiosis and form clonal gametes. However, interestingly, that those eggs, like just activated by sperm, as I told in the very beginning, by sperm of other sexual species and start to develop and clone themselves. And when we started to investigate the Korean complex, we expected something similar, as this complex also includes 2 sexual species, diploid and triploid hybrids. And in diploid hybrids, we indeed found the same mechanism, premeiotic genome endoreplication, so I will not go into detail here. However, in triploid hybrids, we found in diplotin stage that there is no orange genome, and these 2 blue genomes, they form bivalent, very similar to sexual species, with no premeiotic duplication, and just simple bivalent, similarly to sexual species. In pachytene, we see crazy things, even more complicated story with repopulation of cells, but only those cells which was able to remove the orange genome is going to diplotin and is able to form gametes, but all others, they are just vanish during apoptosis. And similarly, we detected the ploidy of germ cells, and we found that indeed there are 2 populations of germ cells. Some of them with initial ploidy level triploids, but some of them are diploids, so it means that they discarded the orange genome. So this is the mechanisms of reproduction in this complex. And interestingly that here we have drastically different mechanisms of reproduction during gametogenesis in diploid hybrids and in triploid hybrids. While diploid hybrids exploit premeiotic genome duplication, triploid hybrids exploit premeiotic elimination of single-copied genome. While 2 remaining genome, they can form bivalents, so it's like here, form bivalents and form haploid gametes. But in this complex, in contrast to European one, sperm is always needed to fertilize the egg, not just activate it. So based on this, we suggested a scheme of reproduction of those hybrids in natural population system. It might look very complicated. So after crosses of two species, diploid hybrids appear. Diploid hybrids produce diploid eggs, and they're fertilized by sperm of blue genome, and resulted into triploid one. And triploid one remove a single copied genome and form haploid egg with only blue genome. And these eggs must be fertilized by orange genome to create a diploid hybrids again. So to have things a little bit more general, we starting to search for commonalities between different asexual species, and we compared naturally existing asexuals between fish species, amphibians, and reptiles. And we found interesting, some commonalities between all these asexual vertebrates. And first of them is that with just few exceptions, they have gomomorphic sex chromosomes. And another thing that they emerged after crosses of parental species with certain genetic divergence. So there is a kind of window of the divergence between sexual species to create those asexuals. And we decided to test these natural cases on our model organisms. So we selected cobites, inhabited European rivers, and those species have different genetic divergence, and which is a kind of good for us to test it, and also have different karyotype morphology. And this is a scheme of resulted crosses. We have some successful, some not. But coming to the conclusion, we found very different outcomes, and also here we partly include the existing literature data. And first of all, we found when genetic divergence between crossing species is pretty high, hybrids are inviable, which is expectable. On the other extreme, going to green one, when crossing parental species are too close genetically, they have meiosis and no clonality. At some point, we have, when genetic divergence is pretty high, we see sterility in both sexes. However, one of the most major outcomes of our crosses was sterility in males and clonality in hybrid females. And this clonality, it's also really have this window. So indeed, clonality is not so rare, at least within cobites, as might be expected. An interesting thing that we found that not only genetic divergence can play a role in the emergence of clonality, but also chromosomal similarity. Because when we took some species which have diverged, more different karyotypes and closer genetic different karyotype and closer genetic distance, they have clonality. So there are some crosses like this. However, when karyotypes are very similar and genetic distance was sufficient, they have meiotic reproduction. And we did next step. So once we have those hybrids, we cross them with parental species to make back crosses and obtained triploid progeny. And interestingly, after brief analysis, it's still ongoing project, that at least in this 4 combination, we see the same situation as in Korean one. So we see elimination of single copied genome and the formation of haploid gametes between double copied genome. So I would like to conclude that clonality, at least in cobites, is really not rare and widespread outcome, even in laboratory crosses. And there are 2 gametogenetic operation, duplication and elimination, which appears instantly. And taking into account that there is some genetic distance required between crossing parental species and clonal genome propagation, clonal genomes, during their propagation, they're isolated from each other. So we can speculate that asexuality may be a kind of type of postzygotic barrier for for at least lower vertebrates. I would like to acknowledge my colleagues, collaborators, and funding source and organizers, and you for your attention.
Yes. I might have missed this, but does this occur naturally in the wild, or is this only in your lab?
Well, both. Like we have a clonal hybrids in nature between different lower vertebrates group, but we decided to use our model to recreate it, and it works as well on those species which were never crossed in nature.
Hello, bonjour, everyone. Thank you for being here with me. My name is Luis. I am a postdoc in Concordia University here in Montreal, but today I'm going to talk to you about the last work that I did during my PhD in Mexico. So in some hybrid zones, we can identify these kind of patterns in which we see gradual changes in genotypic or phenotypic characteristics across the hybrid zone from the allopatric populations of one species to the other. And genomic lines framework allow us to describe these changes, these gradual changes, as a combination of two evolutionary processes happening simultaneously. So on one hand, we have selection against maladaptive hybridization happening inside the hybrid zone, but at the same time, we have the gene flow of individuals coming from the allopatric populations into the hybrid zone. And the intensity of these processes, it is what shapes these changes in the phenotypic trait or genotypic trait of interest across a geographical distance. And analyzing genomic lines in hybrid zones is really interesting, because if we do it for several loci at the same time, we can begin to identify patterns, the neutral integration that happens across most of the loci, as well as outliers in which the clines behave differently. And this is really important because these outliers can allow us to identify loci that are under selection, either that they restrict the gene flow or that they can have an excessive integration from one species to the other, and so may be related to processes of adaptive integration. And an extension of these client models are the so-called genomic clients, in which the geographical distance of the models are shifted to the indexes such as the hybrid index, which measures the ratio of alleles that one individual have coming from the parental species. And so the genotypes of each individual are modeled as a function of their hybrid index. And so these so-called genomic clients allow us to incorporate the methods of the client framework into mosaic hybrid zones, which are hybrid zones that do not behave as the traditional tension clients in which the first clients were described. So. And mosaic hybrid zones are interesting experiments in nature, because if we sample patches in these mosaic hybrid zones where the two species live in symmetry, we can begin to test the consistency of integration and hybridization outcomes. And so the main questions that I wanted to ask was how consistent are genomic clines pattern across multiple hybrid zones of the same pair of species, and how the specific demographic history of each zone may or may not change the observed genomic lines. And to tackle this question, I did an empirical work studying a mosaic hybrid zone formed in Spain between two damselfly species. So here in this animation, I am showing you in yellow all the observations of one of the species, the native one, Ischnura graelsi. And in blue, you can see across time the field observation of Ischnura elegans. So Ischnura elegans went through a process of expansion of its range of distribution, moving westward into the Iberian Peninsula and entering in contact with Ischnura graelsi. And in this system, we know, based on measurements of reproductive barriers, that processes of reinforcement of reproductive isolation may be happening in sympatry. And so our objectives were to test the independent formation of Ischnura elegans and Ischnura graelsi hybrid zones using demographic inference analysis, model and compare genomic lines between the hybrid zones, and then identify, describe, and compare genomic lines outlier lossy between the different hybrid zones. So here I am showing you the map of our sampling localities. And so we went to the field and we sampled organisms from the allopatric populations of both species, as well as sympatric populations of preliminary called 3 hybrid zones. So we have the western hybrid zone in Galicia, northern Spain, the central hybrid zone in green in La Rioja, mainly La Rioja, and in orange, the eastern hybrid zone across the Mediterranean coast. Then using these field samples, we did conventional sequencing for population genomics using RADSEQ, and we called 2 kinds of SNP variants. So we have the full SNPs variants, almost 7,000 SNPs from the RADSEQ, and also we identified diagnostic loci, which are SNPs that are fixed for one allele in the allopatric populations of one species and the alternative allele in the allopatric populations of the other species. And so all my analyses were done using both of these data sets. And so first here I am showing you a structure analysis, specifically PCA, but all the other methods show consistent results, in which on the left side of this PCA, in the principal component 1 axis, we see the Ischnura elegans samples, in the right Ischnura graelsi, and so it allows us to identify the 2 species as well as hybrids and introgressed organisms from nature. And interestingly, we see that Ischnura elegans, in the recent time that it came in Spain, has begun to form a genetic structure. So we see the allopatric localities forming a single cluster in blue. In purple, the individuals from the western hybrid zone in Galicia, and a third cluster formed between the central and the east hybrid zone. Meanwhile, Ischnura graelsi, the native species, lack this structure and acts more like a panmytic population across Spain. And then we used do-it-yourself approximate Bayesian computation to model and test different demographic models. And specifically, we were interested in testing if these hybrid zones were formed by independent admixture events. So as an example, here I am showing you how it looks with 2 hybrid zones, take into account that we did for the 3 areas. And in the top row, I am showing you 2 models for independent formation, so 2 independent admixture events. The only change is the order in which each hybrid zone was formed, while in the bottom row, I am showing you examples of secondary formation. So models in which first a hybrid zone was formed, and to migration, individuals formed the second hybrid zone, again with differences in the order of appearance of the sympatric regions. And here I am showing you the most probable model, in which it selected as most probable the one in which the 3 hybrid zones were formed by independent admixture events. Another key difference is that in the east hybrid zone, the genetic contribution of Ischnura graelsi was very low, when in the other 2 were more homogeneous between Ischnura elegans and Ischnura graelsi. And another important result of this model is that it shows that these events happened in very recent times in evolutionary scale, so just a few decents of generations ago. And these results are consistent with the field records, with how we see this process happening in nature, because you can see that it was around the 1980s when the real boom in observation of Ischnura elegans began to happen in Spain. And so then with this data, we began to do our genomic lines analysis. And to do this, we decided to merge together the samples from the central and the east hybrid zone, mainly based on our results of the structure in which we see that they form a single cluster with the Ischnura elegans samples, and also to have a bigger representation of individuals across this gradient of hybrid index. So now it looks like this. And after modeling the genomic lines, we began to see that they have different patterns of how the cleans behave across these two hybrid zones. So we took, or we estimated the steepness of these, each of these clients, both the neutral and the outlier clients that we detected, and we compared the steepness or the rate of change across the two hybrid zones. And so we did this genomic clients modeling using 3 softwares, and in the 3 of them, we consistently saw that the central plus east hybrid zone had clients with higher steepness values, so the integration happened more abruptly, while in the west region, which is the older, there seems to be low or smaller values of client steepness, suggesting higher integration in the west hybrid zone. To confirm this, we did a BABA test, and we compared to test for significant integration in both hybrid zones, and we detected significant integration happening in the west hybrid zone as a higher frequency of ABA pattern than BABA, while in the central hybrid zone, we did not detect this kind of significant pattern. Then we took out outlier lossy from the client analysis, and we compared them between the hybrid zones. And interestingly, we, in the 3 different software that we use, we identified that most of the outlier lossy were not shared between the hybrid zones, and also that there were a neutral lossy that behaved differently, significantly differently between the 2 of them. Now I want to focus the attention on GG hybrid software. This is a very new algorithm to detect this outlier lossy, and it has the extra addition of identifying or suggesting the selective pattern that may explain these outliers, and so they classify them between restricted integration or excessive integration. And we compared the relative frequencies of these outlier classes between the 2 hybrid zones, and we detected that there is excessive integration or higher frequency of excessive integration outlier lossy from one species to the other in the central hybrid zone, maybe supporting recent evidence of adaptive integration that could be happening. But in the west hybrid zone, we detected higher relative frequency of outlier lossy related with restricted gene flow that might be related with the reinforcement that we see in this region. So as conclusions, genomic lines modeling show us different patterns of integration. These integration patterns and outlier SNPs could be related with the demographic history of each hybrid zone, as well with asymmetric speciation processes that we have described that has been happening in this species in our other works. And I just want to finish my talk saying thank you, gracias, to all the people that work with me in Mexico, and especially to my advisor, Rosana Sanchez-Guillen, and thank you all for being here with me and listening to my talk. Okay? Okay, so thank you.